for the last lecture of today, I want to start with part A of what we call experimental design, which we'll continue tomorrow. And today it will be kind of a bit conceptual, whereas tomorrow we'll focus more on the actual practicals, on the equipment and how all these things work. This one. So the outline is that we'll try to start out with a broad reflection on how we can understand, use EEG to understand the relationship between mind and brain. And this is rooted very much in cognitive science, but I think it has a relevance to also think this into a clinical setting, because I'm not so fond of this whole distinction between you are at the cognitive department, you are at the clinical department, but I think in the end, we are studying how the brain works, or sometimes how the brain doesn't work when you get diseases. And this is kind of, rather than being opposite fields, it's kind of tangential fields that you can have the normal functioning, you can have abnormal functioning, but we still need to think of how we think this function into how we approach brain science. I will kind of start to outline what we should look for in our data when we think of our experiments. And very much a point I like to stress here is that when we design our experiments, we have to think of already there what is actually the end goal, what do we want to get out of our data. And then I think I will end up with some example of what we can conclude, and I think even more important, what we usually cannot conclude from EEG data, some typical errors that I see people make. Going back to this whole setup scheme that we have right now, I want to focus mainly on how we use a task together with EEG to study brain function. So I showed you this earlier today, that the central problem with neuroimaging is that we want to understand something inside the head, but we're standing outside and now have to measure something, and we're measuring EEG in this case here. So the logic of this, having kind of like a black box approach, is that it's actually nothing new. It goes all the way back to the foundation of psychology and cognitive science, in that we would study some behavior in an animal and we would present it some stimulus, then it would behave in some way and give some response that we could measure. So in the very old school behavioral science, it could, for example, be how punishment and reward would affect a mouse behavior. So we'll give it either a small food pellet or an electric shock, depending on how it did on a task. And then we would see how this behaved. And from those kind of line of research, we got basically the concepts of reinforcement learning and Pavlovian conditioning and so on, so that how reward and punishment would affect. Sort of later on, and I think this also coincides with the bump on the graph, so that in the 60s, like cognitive science started to emerge. And in hindsight, it might not seem so revolutionary, but it was, rather than thinking it's not just sort of stimulus response, that one-to-one mapping. There was something more going on, and they were showing that different kind of stimuli could be processed in different ways. So many of these, what this schematic is trying to illustrate, is to start to come up with some hypotheses about how different modules of function inside the mind would actually process the incoming stimuli and then lead to a response. So cognitive science 101 here. And I think for including the brain into this scheme here, isn't actually conceptually that different, because we're still measuring something, usually some response to some stimuli, and then rather than now just hypothesizing about what's going on, we're trying to measure what's going on inside the box now. But we're still standing on the outside and have to make some decisions on how we interpret the data that we get in the end. And also like to think this idea that when we think about responses or behavior, having been at a hospital, there's a very clear distinction between what people say like a neuroscience measurement and a behavioral measurement. But if we look just at studying an organism, this organism being a human being, Like we can define behavior as many ways. We can think of behavior as how people just behave out when they walk around in the city. We can think when we are in an experimental setting as behavior as reaction times and performance and so on. We could also think behavior as, for example, a reflex muscle behavior or something. And now we're actually starting to move into physiology and even neurology. So we have the peripheral reflexes and so on. And I like to think that behavior also could encompass actually the behavior of the brain. So part of the human is responding to something, here particularly the brain, exhibits some certain behaviors, now here expressed in voltage or oscillations and so on. So basically we have some stimuli we can apply to our participant, and we have a lot of different readouts we can use to try to infer what is going on inside the head. So basically we've not really changed the game of how behavioral science or cognitive science work. We have just now added brain data. And I'd like to borrow this metaphor from my former supervisor on how to understand what we're looking for in the brain. So as I showed you in the morning, that we are measuring from the scalp, we're measuring basically the whole brain at the same time, but we're usually interested in some specific responses to our specific stimuli or related to the concept or the disease we are interested in. So it could be a bit similar to imagine a big classical orchestra that is playing some music. And they're all playing in symphony, and similar to the brain, it's not just completely random, it's actually synchronous. They have to be very much in concert to play. But maybe we're only interested now in understanding the specific, how the cellist is playing. So we could think of that similar, we're interested in the visual cortex. But the problem is that we are standing outside the concert hall with a tape recorder. And now we need to find a way with this recording that we're doing to try to isolate the specific function in the orchestra that we are looking for. And going back to kind of the very usually considered the birth of cognitive science, Francis Cornelius Donders. And if you heard the name Donders before, that's the university that Robert is from is actually named after him. So he was already, I guess, 200 years ago, 150 years ago, came up with this idea that actually has been the very, I think, fundamental in both cognitive science, but also in neuroscience, even perhaps without we know it in that. This is what has been known as the contrastive method. And in this experiment, I think how he did it was quite simple. It was a simple go, no-go task that some of you might have seen before. It's very easy. It's basically the participants are sitting, asked to wait, and then at a certain time there would be like a signal that said go. In modern version, it might be that you get like a green light that say, and then you just press a button or move your hand or something as fast as possible. So the idea is that we have some stimulus, a go response, a stimulus, the go cue, and then a response. So very classic behavioral setup. But in some other conditions, the instruction would be the same. So the participant would sit and wait. But then in some trials, most trials, there would be the same green light that would come on, and people just have to respond as fast as possible. But in some trial, actually a red light would flick, which would say you should not do anything now. And then again, they should not give a response. And if you try this, sometimes this can, if you're really ready for things, you actually accidentally respond here because your system is so ready. But what he then showed with this is that looking only at all the cases where people just have to respond as fast as possible. So they had a certain reaction time when it was just like the very simple task. But on the trials, which should be the same, that they just should respond as fast as possible, when there were this kind of interference of the no-go task, people's reaction time were slower. And this you say that based on the reaction times, that the behavior is consistently different. We could see that the difference here, that there is a difference between the two tasks. We would infer that there is some other cognitive process going on in addition to what would going on in the first. And now, 150 years later, we usually say that this would be like an inhibition response, that you are inhibiting your impulse to act as fast as possible, because there might come some interference. And all this is kind of inferring what is going on inside the head just from some behavioral data that, in this case, very simple reaction times. I like to have this one, the Stroop task. I guess some of you might be familiar with it. So the task is simply that you say the color of the word. So not what is written, but the color of the actual letters. So you can try this just for yourself now. Yeah, it's quite simple. So now I want you to do the same. Say the color of the word on the words that come up over here. So say the color, not what is written, but the color of the letters. Here you can really experience this like interference effect yourself, right? A lot of you were laughing. I am horribly bad at this task, so I'm not going to try it either. But it really demonstrates the logic in this. So a bit more formal, if we were to look at this, we might have sort of a theory about a function, like how different, in cognitive science, we like to just set up as these hypothetical modules. But I think when we think of it in terms of neuroscience, these modules could be like areas of the brain or processes in the brain that are interacting. So we could have a theory about some kind of architecture that would facilitate this, this process. And that we could say, maybe we say if we process some information, it would go in this way, but there would be some module that would kind of monitor whether there's some kind of conflict. And this detour would make you slower on those where there is an interference, and that would predict that when there's no kind of mismatch, you are just faster at those trials. Then we could have, for example, another hypothesis, sort of proposing an underlying architecture that would say, no, it's basically just the same. They would be equally fast. It doesn't really matter what way it is. So now we have two hypotheses that we can actually test, hold up against each other, and we can then test which of these would, like empirical prediction that we make, will be faster or you'll be equally fast, and then hold it up against our test. So it's a very simple hypothesis-driven perspective here. And that we can do with reaction times, but we can also use brain responses to actually test this. This allowed us to dive into new kind of avenues that reaction time, perhaps reaction times are very crude measures, but now we can look specifically perhaps at some of the subcomponents in the brain networks that we are looking at that can start to answer some of these cognitive questions as well. So an example here from actual EEG data that uses this kind of contrasting methods to try to understand brain. I think in this experiment, the participant was sitting, listening to some beep sounds in both ears that would come sort of alternating left-right. And once in a while, there would be a beep sound that was kind of an odd one out, so at a different tone than the rest of them. And the participant was then asked to count all these like odd one out tones, but only pay attention to one side, so one ear or the other, either the left or the right. And what is shown is this is that when you just look at all the different sounds at the N100, so here around 100 milliseconds, it's very typical you have this negative peak associated with sound processing, that there was a much stronger response for that when people were paying attention to the tone versus those they were not paying attention to. So here's showing that you might not get any behavioral response, but from the brain response alone, we can see that attention actually modulates the brain response. And I think the other one here is then just how are the difference between sort of the regular pattern versus those tones that are standing out. That they show this later in the processing stage to see that there is a large difference between those that are called the standard towns and the kind of target that they have to attend to, saying that this P300 effect was more involved, I think, in the monitoring of the target that they have to keep track of. So that is one way that this contrastive method is used in EEG to basically look at the difference between conditions here. And this, I think, is a very powerful approach that we have in EEG to isolate sort of different components over time here. So cognition is also kind of very broad. There's many ways to approach it. I kind of divide it into three categories here. You can think of it as sort of basic concepts about cognition, which could be very basic, like attention, working memory, decision-making, reward processing, or these kind of things. You could also think of a cognitive model, which is more in a formal way, trying to say how are these different concepts made of different subcomponents that would explain the function of these. So this is, I think, a very famous old model of working memory, where you have like sort of different subcomponents on the responding to different domains, and then you would have like a central buffer that kind of maintain these. I must admit I took this from my psychology textbook. I'm not so well versed in the newer development in memory research, but just to illustrate that that will kind of predict some interaction between different models. And what we then can use EEG for, and neuroimaging in general, is to try to understand these concepts, how they are rooted in the brain. So if we have like a basic concept, like for example attention, so in this case here, we could see just how attention affects a brain response. We could also test some specific predictions about a certain model, saying that if we change some module here, the brain would respond different. So in some come up with some task that would target some specific part of our cognitive model, and then we would see whether would we actually see a difference in our brain data. Cognitive modeling, I think, is a kind of different way. So I think I picked here one of the most complex diagrams I could find, just to show how it can be done. So in this experiment here, they are building on this predictive coding framework. If you're not familiar with that, that's perfectly fine. It basically means that the brain is constantly trying to predict what is going on. And it does that in some hierarchical layers that will try to, that constantly interact between each other. So I like this particular model here, that because they started out with what they thought was, or what they think are some physiologically informed ideas about how the brain should work, to create kind of a cognitive model that still have this very much flow diagram type of thinking of cognition. And in this particular study, they actually tried to mimic how the brain would respond to oddball. So in this having a standard tone and then an odd tone out, which gives rise to a very particular brain response, and then try to simulate, given this, would we then see some responses that look like an EEG response? And in this here, we have kind of a two-way interaction between the theory that you can think of these mathematical models as a theory of how the brain and the mind works, that then would try to explain some phenomena we would measure. In the very simple, it could be reaction times, but here it could also be some brain responses. But in the same way, what we measure with EEG, for example, well, this theory, if we buy all the premises and the modeling approach, could then potentially explain what we measure with EEG. So that would be some way to think how EEG could inform cognitive theories at different levels. And I think now I rooted this in cognition, but I don't think when we apply this to like a clinical setting, it's so much different. So we could have here basically that maps kind of one-on-one to the clinic. We could have a patient group who have reduced attention, and we could then basically see how are the responses in the brain different from from one group to like patient group to controls. We could also have some theory about some mechanisms that break down, for example, with like motor control in Parkinson's disease, that we would say there are some specific pathways that between the basal ganglia and cortex that are breaking down or are not used due to the lack of dopamine in Parkinson's disease. And we could set that up in kind of a diagram and that, and then we would try to see, well, how does this then map out differently? If this is the case, would we predict something that we could measure in the brain? I think this approach is starting to get kind of adopted in clinical setting, or at least in basic clinical science. I know there's field of computational psychiatry that try to use models like this to sort of explain how different diseases, for example in schizophrenia, might why there are some like these more fuzzy symptoms that they have. It's not a field I'm super familiar with, so I will not dive more into detail on that. So that is how sort of conceptually I think that EEG and neuroimaging in general can really inform about the process of our mind, thinking about these three levels. So we have this kind of, on one side, our understanding of cognition, and the other side, our understanding of brain. So how do we then translate this into something practical that we can actually measure? How do we combine these measurements? So basically in the beginning, we think that there will be some link between some cognitive theory and some process that we will be interested in studying. So let's say some visual perception and some specific process going on in the visual cortex that we're interested in. But in order to study this, we then need to come up with a cognitive task, a task that we know would tap into the process that we're interested in. So if it's visual attention, we might have a task where the participant actively have to attend to something and maybe suppress something else. And from this task, when they're doing that, we would then usually get some behavioral responses that we'd be interested in. That could be like reaction times or performance and so. So we have this whole behavioral column of things we could do. This experiment we could in principle do without brain measurements, but now that is why we are here. So when it comes to the measurement, we also should think of how do we capture this brain process? And since this is an EEG workshop, that answer is kind of given. We want to use the EEG, and I will focus on that. But going back to this morning, there might be some questions where different scanner modalities are actually just better suited for that. And I think for EEG, what we really are capturing is the high temporal dynamics, and having these waves, we can see how the brain responds to stimuli. Whereas if the question is where in the brain is this process going on, fMRI is perhaps a much better method for that. If we're interested in how neurotransmitters are involved in this task, PET measurements might be what we're interested in. So ideally, we'll start out saying that we find out how do we best measure the brain process we're interested in. I know in reality, it's not that I can just go into the PET department and say, I want to scan a lot of participants. So sometimes we're also a bit limited to what equipment we have available. But the thing here that we have to think about is how do we actually capture this brain process that we are interested in? Yeah. And then in the end, we get something that we actually record. And in case there would be like our stream of EEG data that we would then, in the end, try to link back to how both our recorded signal inform us about the brain process, that we are actually tapping into the process we're interested in. The same that can say at the cortical column over here, that we want to make sure that our observed behavior is actually an expression of the underlying cognitive phenomena, that we can say this internal validity of the study that we are conducting, that we are actually picking these things up. And in the end, what we are then asking, so we have some cognitive tasks we might have defined here, and some brain measurement we have here. is in the end, is there a correlation between the changes that we see here that varies according to how we think the brain is actually measuring or processing things to begin with. And it basically means that when we have some brain measurements, we usually talk about the brain correlate of some function. So the brain correlate of attention. Because we cannot do it the other way around and say that, oh, we see this difference between attended and unattended. Therefore, this must be where the brain is doing attention. Because, as you know, we're always measuring a lot of things, and processing is not just a matter of one place in the brain. This is usually what refers to as reverse inference, and that is not really logically sound. So we will not actually be able to make these kind of conclusions from our data in this strong causal sense that we can look at the brain data and saying now the brain is doing this. Think of this in the same way as a concert metaphor, that everything we measure is part of this whole concert. The cello player is in there somewhere and might be playing extra loud right here, but they were playing all the time and all the other was also playing. So the type of things we can kind of conclude from something like this is that we can conclude that attention, since there is a difference here, is clearly involved around this 100 millisecond time period. But even better, we could also test some specific hypotheses about how attention is involved by having a carefully crafted task that would tap into some attentional mechanisms. And then we can look at kind of the difference between different cognitive or brain functions. So here just denoted like FFF, that sort of could be any function we're interested in, given the kind of task or condition that we are measuring these under, which is like X1, X2, and so on. So the brain is a big puzzle, and we kind of have to find out how our pieces fit into this here. All right, few time for some question if you have. I know this is, yeah.
Very much. You said that under the brain, under the measurements are made of, neurotransmitters can be measured.
So yeah, not with EEG. So that was an example of how we could use different scanners. Yes, so that was good. So the example there was that if I was interested specifically in neurotransmitters, which, for example, if we're working with Parkinson, where dopamine is relevant, I would not be able to measure that with EEG, but that I'll use a PET scan for seeing the receptor density. But I could have an idea about that we know in advance that Parkinson patients have dopamine depletion, and we could get perhaps some indirect measure of that, like medicine dose and so, and we could use that as a way to look at our EEG data, for example. So while we are not measuring it, we could have it as an indirect kind of measure in our study. Yeah. So talking about experimental designs, this is kind of not that unique to EEG, but if I had to give the EEG spin on it, that it would be very common in kind of this kind of task. We'll try to isolate some function to have a within participant design that we call, which basically means that for all the different experimental conditions that we compare, every participant in our study would do all of them. So in this case here, it's from one participant, and we would just repeat this task in all the different participants that we will have. So having attend to one, attend to the other, and so on. So if we see that we have our research participant A, participant B, and how many we need, the comparison is kind of between task A and task B. From an EEG perspective, there's a strength in this is that we can kind of control or like control a bit for the noise that will be in the EEG measurements. So different participants might move to a certain degree, blink to a certain degree. And between experimental conditions, we want these things balanced as much as possible. When we're doing a within participant design, those things tend to balance themselves out in the run. So I account for person level noise. Of course, this is not always possible. For example, if we're doing comparing different populations, so like a clinical group versus a healthy group, well, we kind of are comparing between participants. So between participant design are also happening. It could be then the same task that all the participants are doing, and then we will group all the healthy participants, so in the person A group or person B group, and then compare those two groups with each other. There's nothing inherently wrong with that, but we have to be aware of between group noise, saying here. And from my own research on Parkinson's disease, there are some clear differences in these physiological artifacts that actually differentiate between patients and controls. For example, there's a tendency for Parkinson's patients to have these more stereotypical face mimic, which means that they tend to not actually move their facial muscles so much and blink a lot less. So they're actually really good EEG participants. Yeah, and but that means that actually the kind of level of noise are different between the groups. So if I want to study a certain function, I have to make sure that I have cleaned this data to the same degree that I'm not accidentally concluding a difference in brain function, which was due to some trivial noise component. So I think a good way to approach this could also be in a mixed design, and I think these are they're called mixed design when you read about them from psychology and so, but I think they're very common in neuroscience, which is that you have basically different groups, but within the groups you also have different conditions. So it's kind of balanced that you have in the green group and the blue group, they're both doing the same two tasks. So it's basically a mix of these two here. Because then we can look at the difference between the tasks to kind of isolate some effects, and then those effects we can compare between groups. And I think where this, just an example of where this is used in EEG, is this classical mismatch negativity, which is again this task where you have some tones that are played, and once in a while there's a tone that's different from the others. And when we can just measure the brain response, so the tone is played here at the time S, and we see, I think, in the solid line is this the most frequent sound, so the standard it's called, whereas the dashed line here is the kind of the odd one out. And we can see here that they are different, the two waves, but what is typically done is to look for what is called this mismatch negativity, which is defined as subtracting these two waveforms from each other, and then we get this like nice component going on here that then reflect kind of the difference in the processing between some standard high predictable and something that basically stands out. And this is a very well-known EEG feature. And this method of kind of subtracting two waveforms from each other is usually referred to as a difference wave. And it has some strengths when we also want to look at what underlying processes are actually going on. So as I told you earlier, it's difficult to infer processes from scalp potentials alone. We could look a bit of the topography and try to guess whether there was a dipole pointing in some direction. But when we just look at a waveform at the scalp like this, as I said, there could be many different underlying configuration of sources that will give rise to this. So in this diagram here, let's say A up here is what we measure, and we look at some different peaks in an ERP. But B and C here would represent two underlying waveforms, so a a combination of three sources, C1, 2, 3, that would then, when summed together, give rise to this waveform. But it could also be what is shown here, so they call them C1 mark, C2 mark, and C3 mark here, would then would also sum together to give this observed ERP waveform. So just looking at the peaks alone, we would see that the peaks doesn't necessarily line up with the peaks of the underlying components, and depending on which direction this kind of goes, we would get some like these different waveforms here. So if we compare this, I think having sort of like a first component, then a longer sort of sustained, and then one kind of skewed very high here. But because these two are going in opposite direction, it looks in the measurement as if we have a sort of short peak followed by a positive increase, when in this case down here, it would be a result of two overlapping opposite going components. That also means when we look at the waveform alone, it's not always easy to infer what the underlying process is in this case here, because the scalp potential doesn't map one-to-one to the underlying sort of neurocognitive architecture. So in the example of D up here, it would be this. That was the kind of true underlying configuration. And then we have two experimental conditions. And in one of them, we see a decrease in amplitude of C2. So this would be more towards 0. But when we just look at what we would measure at the scalp here, so the dashed line would represent the second condition where this is reduced. We see actually there is an increase, like the second peak kind of disappear, and the third peak kind of both shifts a bit towards earlier in time and would also increase in amplitude. And if I didn't know better, just looking at this, I would conclude, yeah, we are losing the second component, and then we have a strong increase of the third component, when in fact it would just be like a single component that is changed here. And the same example in E here, now we are in this case here, where we then see an increase in the first component, which because of the summation of C1 and C2 here, gives kind of this increase in the peak of the first one, but we see that the latency is also pushed a bit further back. So we might be tempted to conclude that in this case, the participant was slower. And then we see that this affected also the second peak. But in this example here, it was only the very first component that changed. And they're kind of the same in the third condition. So here it's just the third large one, and the lesson is kind of the same, that we see that the waveform changes in many ways, but that it still could only be one underlying component that explains all of it. So the way from this is from the book I recommended, they said we could isolate these kind of things, was to subtract the two waves from each other and then look at the difference here. So in this case that I thought of, we could see that it's actually able to kind of recapture the true underlying components. That is, of course, here in this case, based on the assumption that there is only one component that's changed. In a real experiment, there's probably more things that change along the way. But it would be a way to, rather than rely on trying to find where the peaks are happening, to actually give a good estimate of where is, like, what is actually the difference between these two. So if we have a task where we control basically as much of the surrounding variables as possible, that we can manipulate some specific functions we're interested in. With this contrastive method here, by calculating difference waves, we can kind of try to isolate what is the difference that makes what is the difference in the brain activity that makes a difference from our for our cognitive process. Yeah, so good matching conditions, and then we can subtract waveforms. And the different waves is kind of easily defined as just having condition A, waveform, and subtract condition B from that. So it's mathematically quite simple. That leads to kind of in an experimental setting that ideally, when we have two conditions we want to compare, the only thing we want to differ between these two conditions is that one function that we are interested in isolating. But of course, in reality, this can be quite difficult because there will be some confounding effects in the environment. But as an example, for example, I want to understand visual attention. And I want the only thing that really should be different is whether people are paying attention to something or not. This would be a very simple experiment. But there's still many factors that potentially could go in and influence the brain measures that we measure. So some common experimental confounds that I think are very common. So and we should definitely think about when we design experiments. This is not a comprehensive list, but I think the most common would be that the stimulus features are the same. So for example, if the example before I wanted people to pay attention to one sound or another, I should make sure that at least the sound intensity is the same in both ears. So if one is much louder than the other, it might skew the way that the participant do the task. I think also the engagement in the task makes a huge difference. So for example, if you have a task you want to compare where one is super easy and one is very, very difficult, you suddenly have a lot of other confounds in this that requires, for example, a lot more cognitive engagement, which will also engage other parts of the brain. So we might not be able to isolate. Attention plays a huge role in EEG. So we saw that many of these components and so are influenced directly modulated by attention. So for example, I think this a good example would be if you're comparing two different groups. If, for example, you have a ADHD group who are very easily getting distracted and a healthy control, well, if we want to see whether they are processing visual stimuli in the same way. If one of them are not paying attention, they're not really doing the same task anymore. Another thing is actually behavioral responses. So if you, for example, take a task where people have to respond fast, well, if we have, for example, comparing young adults with elderly participants tend to be slower, which means that the reaction time here might actually go in and influence how the brain responses are as well. So that's something we should think into our design as well. We could, for example, have them not respond right away, but respond at a later time, or in some way incorporate the reaction times into the analysis. So I wrote some possible solutions here, but I also want to stress that there's not really one solution that fits all. And sometimes we just run into some experimental designs where we cannot isolate these factors completely. So say I want to do a reaction task time with young participant and elderly participant, well, this will always be a thing we should think into our design or in our analysis. Or sometimes we just have patients who have reduced attention and we just have to either come up with a task that kind of accommodate that, but then it might be too easy for the healthy participants who would then lose attention because they would start to think about everything else. So sometimes it's a problem that cannot be solved perfectly, but we should at least think it into our design of the experiment, how these things would. There's also some more common EEG-specific confounds, and that could be blinks. So if you have some participant that blinks a lot versus some that don't, I gave Parkinson patient as an example before. One way to take this into account could be to try to control when people blink. So have like a screen before a trial starts that say, please blink now. Then people would blink a little, and then maybe their eyes are wet enough that they won't blink during the trial. So we control that. There's a danger in that also, that if I tell you now, don't blink, don't think about blinking, you would usually start blinking. So paying attention to these automatic processes might also be a confound. I think environmental noise can make a huge difference. We'll talk that more about the practical things tomorrow, but kind of keep a stable setting. And if you have some for example, medical devices or stimulation equipment that we know introduce some kind of noise. Maybe we could design a task that would still actually have the noisy machine present, but would not actually do anything. So we can then kind of isolate the noise between the different conditions. Muscle noise makes a huge difference in the quality of the EEG signals. So there, some participant might be uncomfortable sitting in a chair in a longer time or whatever they're doing. So I would say always do an experiment, ensure that you don't see the muscle noise in the data and that the participant relax. Vigilance, sleepiness is actually a thing that matters a lot for EEG. When people become very tired, the alpha wave, especially occipital alpha waves, goes up quite a lot. And that can really, it's the largest signal I think we have with EEG that comes from the brain, and we could drown out a lot of other things that we're interested in. So that can be to some way accommodated by having balanced designs and having breaks throughout the experiment depending on how taxing it is on the participant. Again, there's not one catch-all solution, but it's something to be aware of. And again, this might also vary between different groups that you might think, so patients and controls and so. Yes, so just a summary that sort of thinking about how we link the brain and the behavior and cognition. So think about what brain and cognitive function that we want to investigate when we start to define our experiment. Think about what is the relation between the task and the function and the measurements that we get. Then we should think about how can we isolate this function, given all the experimental variables that we can kind of tune in our experiment. What would be a good way to isolate this process? And then, of course, think about what EEG response we should look for. So I would just quickly think why I think this is important to think already in the beginning. So before the arrow was pointing from we have our task and stimulus, and we would put over in the analysis, and that would also be how we would analyze the data later on. But I think already when we design the experiment, we should think of what analysis we would do with this and have that inform which task and how we design these kind of things. So I showed you this earlier, that from the EEG data, we have so many different things we could look at. We could look at spontaneous activity, evoked responses, time frequency responses, some specific interaction with tasks in the environment. And depending on which type of these responses I would be looking for, there was some consideration I have to take into how I design my experiment. So for example, asking myself, am I looking at actually an event-related activity or spontaneous activity would determine really whether I would do, for example, like a resting state task or some more elaborate cognitive task. I would think about what location, what latency, and whether I would be looking for things in the time domain or frequency specific responses. That's also kind of the scientific hypothesis that I would be thinking about or formulating at this time. So how does the EEG signal that I'm looking at relate to the brain function that I'm interested in? And here, because there have been done so much EEG research, we could look into the literature and see what had other people that, for example, studied attention found in EEG, and we could start to formulate our hypothesis and start to constrain what we were looking at. So if someone found an effect somewhere before, well, that would be a good place for us to start looking now. And then we can think like, start to formulate not just a hypothesis in the sort of grand scheme about how things are different, but make it very concrete in specifically where would we see? Do we see an effect on a specific electrode site, a specific kind of waveform component that we're looking for? Is it a specific frequency where we see something, or is it more open-ended? Sometimes there might not be a lot of literature on it, and we kind of just have to accept that we don't really know where to look, and then approach our experimental design in that way, open up that we might see some spurious correlations but sometimes that's just how science works. I think to make it another metaphor kind of borrowed here is that we have some dimensions that we can look at the EEG data, and that is time, frequency, and space that we can look at. So time would be like the very typical ERPs that we would look at, like how are things, the responses to a stimuli evolving over time. Frequency, we could look at the specific frequency here expressed in hertz, usually seen as a power spectrum like that. So here we have there would be so like a delta wave that is quite prominent. But we could also combine these, so it's kind of like a three-dimensional system and looking at how frequencies change over time. So what we call time frequency responses. When we think about space, so earlier I said that we don't have so good spatial resolution with EEG, but we can actually start to, for example, see whether there would be some hemispheric symmetry, whether it would be one response would be more pronounced in the left or the right. So we can explore these kind of hypotheses. We could also fill out here with some type of analysis I think are less common in EEG, but that could be to actually combine, for example, frequency and spatial location to try to estimate connection either between different areas or between electrodes. That is what we call a more advanced analysis. I think a method like to stress, because it's becoming very popular in the last 5-10 years, and that are sort of classification, multivariate pattern analysis, or non-linear analysis, multivariate, or machine learning is what they kind of often talk as well, where we basically try to use both location, the whole spatial array, and then over time maybe try to classify between different things. So that also falls sort of in between here. So how we want to explore our data, we could think of along these dimensions. Design considerations also, how do we want to stimulate our participants? And basically what this figure show here is a lot of primary senses and how they kind of all project into to the brainstem, the thalamus, and then up to the respective associated cortical areas. So they kind of follow the same logic, and we could do basically any kind of stimulation you could think of. Could be visual, could be some sound we play, it could be smell, tactile, motoric movements, any kind that would kind of lead to this, that then engage this kind of ascending system. I showed you this earlier as well, where we have this kind of strong ascending drive that would reach the cortex. But we could also have responses that are not sort of directly caused by some stimulus, but more like a secondary property of the stimulus we could show here. And that could be more towards the cognitive domain. So for example, if we showed a stimulus feedback, it could be visual, that would say you won or you lost. So the visual stimuli itself would contain some information that would then be processed kind of in a higher order monitor. So these are just some examples of that. In that case, we might have still sort of an ascending drive, but it would not be like this strong ascending volleys. Rather, we would see kind of a loop forming between the respective thalamic relay cells and the cortex. But now, rather than kind of going in one direction, it kind of goes up and then back again and then up again. So now we're starting to see kind of an oscillatory behavior between projecting up, giving feedback down, and up again. And this we might be able to measure. Again, we measure only the pyramidal neurons up here, but that might actually manifest as oscillation in the neural signals that we see as well. So we will dive into more detail in this tomorrow, but in generally we would have, when we measure the EEG data, we would repeat the stimuli, but we would measure it as one continuous stream of data. But the stimuli would happen at some certain interval. So here are some visual stimuli, we would measure it, and they will occur throughout the continuous data stream. So stimulus 1, 2, 3. Then to get the ERPs or the signals out in the end, we would basically average the responses to all the different stimulus, and then we would get this waveform. form out. So let me see if it comes here. We will think about how does the stimulus induce a response. So what type of responses do we expect to this stimulus? What would be the timing of the events that we're looking at? What would be the required number of events that we need in order to get a robust signal in the end after we have processed all our data? I think something that's worth thinking also, is there a repetition effect? So are there some stimulus that when you have presented them many times, people just either stop paying attention to them or the brain doesn't respond to them in the same way? I know people who study emotional responses have a huge problem with this, because you might show them some very emotional picture and they might get a huge response, but actually already the second time you show the same picture, the response is not the same anymore. So there are some very strong repetition effects. One thing also to be aware of is overlapping components. So in a design like that, when the logic of this is that we have some stimuli here represented with the dashed lines, and those will be presented at a certain interval, which we call the intertrial interval. And how to decide what would be a good intertrial interval basically is that we have to think that whatever stimulus we give to the brain gives rise to some stimulus-related activity. And before we do the next stimulation, we want to make sure that the brain is kind of going down to the baseline level. Which means that, try to illustrate here, that at least we should have some window here. And I also added these small dots and kind of say, in most cases, we also want to do like a small random jitter, so it's not completely predictable. And that's basically because the brain is really good at predicting things. So you might starting to get some prediction effect of responses, especially in tasks where people have to respond fast. You see them go into this readiness state before actually the stimulus applies. So you would have, and these readiness states are associated with a build-up in negative voltage that is really measured with EEG. So if people are starting to predict too much what would come, you actually start to have a kind of bias in that. So we would also have some random interval on top of the required interval to make it perfectly balanced. And if we don't have that, so what I tried to draw here is that now we have an intertrial interval that is way too short, so that the induced or the evoked activity here doesn't really go back to baseline before we give the next stimulation. So I drew them sort of a bit shifted here. But since it will already engage a new process here, we engage the same brain areas. And as before, when we have overlapping processes, they basically just sum up. So I did my best to try to sum these up. What would it actually look like? And I think the waveform would look something like this, which is really, you can see, not really representative of the true signal here, but just a mismatch. So this would basically be useless data. So taking care of the intertrial interval is a good thing. So I also borrowed this table because different tasks might require different intertrial intervals. For some tasks, we can just do a lot of stimulations. And for some, we can only do some that are like maybe once every minute. So I think I spoke about emotion as an example where you have, like when you induce emotional stimuli, it actually takes a long time before the sort of autonomous nervous system goes back to a baseline state. So it might have to take up to minutes before we can do a new trial. And that means that if we need a certain amount of trials to get a reliable estimate, well then we just need to prolong the experiment. But maybe at a certain time also people start to get tired, say we need to do a 4-hour-long experiment. Then, well, maybe they fall asleep by the end and we would not be able to. So typically, and I always hear this, that as a rule of thumb, we should have 40 trials. I don't really know where this comes from. Sometimes it's good enough. I would say I don't like these kind of rule of thumbs, because different responses in the brain are varying by different magnitude. So rather it would be good to have a lot of trials, and then we could from that, for example, do an experiment, a pilot experiment where you measure a lot of responses. So maybe you see in the literature that people got like an attention response with 100 trials. Then maybe do an experiment with 200 trials and then try to see, well, do we see the response? Then what if we then do the same calculation with 100 trials or 50 trials to try to get like a more valid estimate of that? But our experiment is also limited really by something as practical of how long can we actually keep the participant inside the lab before they become annoyed with doing the same task over and over again, or become tired or lose focus. Because that is a huge confound, that if you think your participant is sitting and doing a certain task, but they are actually asleep, well, you're not measuring what you think you're doing, your data is basically useless. Design consideration also for frequency analysis. So we could look at different frequencies in the brain, and we will return to that. But I think the most important thing here is that we have sufficient window lengths that we can actually estimate the frequencies of interest. And Robert will go into detail with how we do that on Thursday, I think, Friday. But we should also think about, so this is most relevant, I think, when we think about time frequency analysis, where we're looking at frequency changes at a sort of shorter time scale. But for example, in resting state studies, we might also think of how stable are these rhythms that we measure actually. Because, so again, the alpha waves here, but you have your eyes closed and you see the alpha. But even inside the alpha, you see that sometimes it's more and sometimes it's less, even though it's generally larger than when the eyes are open. And that applies to alpha, but also applies to a lot of the different rhythms that we're looking at. And these things, like how these people do resting state, how the participant actually are doing the resting state can matter a lot. So some people might engage in sort of mind wandering, thinking about a lot of different things. And that would actually change how their brain operate while they are in this resting state. So resting state is not just not going into this default mode, doing nothing at all. Also, when people just have to sit and do nothing over a long periods of time, they tend to be sleepy, especially if it's like in a dimly lit room or so that's actually also something to take into account. We should perhaps also start to think of how the frequency responses that we're looking at might interact with other frequencies. So if we're interested in alpha band activity, for example, in the sensory motor, what is often referred to as mu activity, this has a very particular shape. That means that it has a very strong alpha component and a very strong beta component, which might not reflect two different frequency bands, but might reflect really just the mathematics of how we separate frequencies. Yes. So finally, when we do our experiments, there might also be some secondary, so trying to bring on secondary measurements that we want to take into account. So typical secondary measurements, I think we talked a bit about it when Robert showed all the different things they're doing in the lab. It would be, for example, electrooculogram, electromyogram, or electrocardiogram, if we are kind of worried about that these things might influence in a very systematic way our EEG measurements. So it's not a necessity for all type of recordings, but you could think of in our task, would it be good to have, for example, a readout of some muscular activity if we are afraid that this might contaminate our EEG data. Of course, the behavioral responses when we're doing a behavioral task is usually something that we want to collect and analyze some way. That could be reaction time, performance, accuracy, responses, how people think they rate a certain thing, and so on. We also showed the eye tracker. In some experiments, people really want to control where people are looking doing an experiment. Sometimes I think this is not a really necessity, more could be a nice thing to have. Some experiments might require it. It could be a galvanic skin response. That's, again, I think people who are studying emotions usually have these skin responses as a robust measurement of that, and you could kind of relate that to the EEG activity. Sometimes you might want to obtain video or audio, maybe verbal responses from the participants, so that could also be something. I think in the kind of epilepsy studies I have seen, they were actually monitoring and recording video together with the brain data, and then they could afterwards in the video kind of see here a seizure happened, and then look at the data in that period. So that would be one. It could also be if I think we could add to video also motion tracking, for example, that if people are doing like more complex movement, that's also some data you could combine with that. So all this is like secondary measurement. So in designing the experiment, we also think what would be something that we would need in order to understand our data, and what would be maybe a nice thing to have, but if we cannot find an eye tracker, then maybe we can find some way around it. And this is not a complete list, so different labs have sometimes their own very peculiar way of setting things up, and have some very some weird devices that they use to do some very specific task. Offline measurements. So what we usually talk about offline measurements is what we call here could be participant data. It's generally experimental data that we want, but that we don't really need to obtain while the participant is sitting in the lab and doing an EEG experiment. And this could be something like the demographics data, so like the age, the sex, or the handedness of the participant, whether the participant was a patient or control or whatever we would be interested in. And it could also be, for example, if we want to have some more detailed psychometrics or neurological tests, we don't really need to do them in the EEG lab. We could do those either at the bedside or in a separate testing room, if we have that, where they're not disturbed by all the EEG equipment. That could, for example, be, in some experiments I've been doing with Parkinson, we've usually obtained this standardized Parkinson score. That is sort of a standardized clinical test. And we would do that, for example, before or after the recordings, because that is just, it doesn't affect the recording in a way, but it's a nice thing to have to kind of give a picture of who our participant was. The same way we could have some interview data. If that was relevant, we could have some questionnaire data on some various things that we would be interested in if that was relevant for our task. So yeah, the rule for what you should include of these offline measures is basically whatever is relevant for you to answer your research question. Okay, so that is I think I will stop here. So just a summary of the day. What we measure is the summation of the currents for pyramidal neurons, how that changes over time. And this is an expression of a wider network of brain processes. And we study that by looking at these kind of intricate networks, looking at the kind of top layer in the cortex, particularly the pyramidal neurons, how they behave. But when we look at this, even to say this is what we measure, we should think this into like the wider brain network and trying to see how this can inform how brain process what we can learn about the brain from these measurements. Okay. So I know this was kind of a bit conceptual. Tomorrow we'll go into how the technical side of things we can set this into some actual setups and then try to look at how we would do the experiments. So some question.